So this lecture focuses on vaccines, about the different types of vaccines and how vaccines work. So the learning objectives for this lecture are to recognize the differences between what's called passive and active immunization, to identify the 4 different types of human vaccines, and to recognize the difficulty in inducing cell-mediated immunity and some approaches that are currently being tested to improve the efficacy of T-cell vaccines. So vaccines are an incredibly successful means of manipulating the immune system to protect against infectious disease. And so this is just a bar graph illustrating before and after figures of disease incidence before and after a vaccine is introduced. And you can see it often has a very dramatic effect on disease incidence. So there are 2 main categories of immunization, or types of immunization. So one is referred to as active, and active can be thought of as immunity that's induced by having a natural infection or is induced by receiving a vaccine. Another type of immunization would be categorized as passive, and an example of this biologically is the passive immunization you receive from receiving antibodies from your mom due to antibody transfer across the placenta or because of antibody transfer in breast milk. The other examples are done clinically, where antibody is passively transferred by physicians in the form of polyclonal immune globulins, transferring antibodies collected from one person's serum or a population of people's serum into people that are immunodeficient, and also giving people preformed specific antibodies to deal with specific clinical issues, for example, antitoxins to deal with snake venom. So again, passive immunization refers to the administration of preformed antibodies either to treat or prevent infection, whereas active immunization infers the stimulation of an immune response by the administration of antigens. And this is really what we refer to as vaccination. Finally, there's something that we're not going to talk about today, but you can think of as immunotherapy. And there, that describes the stimulation of an immune response to treat an established infection, malignancy, or other illness. So let's just first talk a little bit about the different types of passive immunization that are used clinically. So one of the jargony terms that we use to refer to immunoglobulin molecules is gamma globulin, and that's because antibody molecules or immunoglobulin molecules migrate at a particular electrophoretic mobility when it's run out in a gel, and it was described initially as the gamma complex of proteins that migrated in that spot. And that complex of proteins was a fraction that contained predominantly IgG. And so what people have done is to purify from the serum of large populations of people gamma globulins, or fractions of the proteins that are in the serum that are enriched for antibodies that are in the population to treat patients with immunodeficiency syndromes. This can also be used to treat people or to protect people against infections that they may encounter prior to travel if they haven't had a chance to get a vaccination that often would take a longer time to induce a protective immune response. One of the drawbacks for passive immunization compared to active immunization is that it's much less permanent. It's transient. It typically only lasts 3 to 6 months. It lasts as long as the half-life is of the antibody that you've transfused into the patient. Passive immunization often involves human-derived antibody preparations. Sometimes these antibody preparations are purified in a way to enrich for antibodies specific for either tetanus toxin or rabies, or sometimes varicella zoster, the virus that causes chickenpox, or sometimes hepatitis B. And these antibody preparations are pooled from individuals that have previously been identified to have very high titers of that antibody to those organisms, often as a consequence of previous vaccination or previous exposure. Sometimes, however, passive immunization involves the use of antibody preparations derived from non-human sources. So, for example, people derive antibodies from horses to get antibodies specific for botulinum toxin or snake venom. And the problem with using those sources of antibodies is that because they're not human, they're non-self, sometimes you'll mount an immune response against the antibody you receive, and it's associated with a specific form of hypersensitivity syndrome called serum sickness. And I'll talk more about different hypersensitivity syndromes in a later lecture. Another example of passive immunization that's used clinically is the production of monoclonal antibodies. And what monoclonal antibodies are antibodies that have a single antigen specificity, recognize a single epitope, usually generated in a laboratory. And one example of monoclonal antibody preparations that are used clinically are antibodies that are specific for respiratory syncytial virus, for infants that are at very high risk for contracting RSV. So that's all I'm going to say about passive immunization. The rest of the lecture is going to be focused on active immunization and vaccination. So vaccination has been around for hundreds and maybe some reports suggest even thousands of years in ancient China. But one of the first recognized uses of vaccination to purposely prevent a specific infectious disease was used by Edward Jenner, where he noticed that milkmaids who had contracted cowpox, which is a virus that's related to smallpox, did not get smallpox. And so what he did was that he purposefully inoculated individuals with cowpox and then challenged them with smallpox and found that they had developed a protective immune response that protected them against smallpox infection. There are different ways of thinking about the immune response that are induced by vaccines. The most straightforward way is really just what we've talked about, how the immune system works in previous lectures, and that's really generating a systemic immune response. And typically the protective immunity that's induced by vaccines involves humoral immune responses and is dependent on antibody production. And so systemic immunity in this case is defined by the production of circulating IgG primarily and sometimes IgM antibodies. And that can be induced by vaccines that are administered subcutaneously, intradermally, or intramuscularly, depending on the particular vaccine. Sometimes people try and target vaccines to just generate mucosal immunity. This primarily is mediated by immunoglobulin A, because as I mentioned, IgA is preferentially produced at mucosal sites and secreted across epithelial barrier cells. But in addition, IgM and to a certain extent IgG may also play a role in mucosal immune responses. Here, what's different about this is that the route of administration usually involves a mucosal site, either administered through an oral or intranasal mechanism so that you're targeting either the respiratory tract or the GI tract. A third kind of immunity that's induced by vaccines is really indirect, and it's something called herd immunity. And what that refers to is the phenomenon by which if you have a population of people that's fairly highly covered with a vaccine, let's say 80% of the population is protected against a vaccine, that'll limit the ability of the microorganism that is targeted by the vaccine to spread in that population to unvaccinated people. Because if you're protecting the majority of a population, it basically reduces the reservoir or potential reservoir of the microorganism, and it limits and it reaches a certain threshold level that results in elimination of the microorganism from the population and limits its spread even to people that did not receive the vaccine. And so, as you know, there's a whole variety of different vaccines that are recommended. In this country, you can look it up and find what vaccines are recommended at certain ages by the Center for Disease Control. And I'm not going to go through these different vaccines. You'll learn more about them in the microbiology course. But there's a whole series of vaccines that are recommended that are given to children at different ages as well as to adults. But here what I want to do is just focus a little bit on what are the features of an effective vaccine, what makes a vaccine effective. And from an immunological point of view, obviously it has to work. It has to protect against a live organism. And the way it does that is by inducing an effective immune response, or the right type of immune response that is effective against a certain class of microorganism. And the other thing that's really important is that it should induce long-term protection, meaning that once you get vaccinated, you don't need to have a lot of repetitive vaccines in order to be protected from the microorganism. And that really leads into the next feature of what could define an effective vaccine. And these are really practical features, meaning that the vaccines that are given should have few side effects. They should not have to be given very frequently, meaning that they require few boosts. Vaccines should be relatively stable, meaning that they don't have to be stored in ways that are sometimes difficult to do in less developed countries, and they should be have relative ease of administration and low cost. Currently in use, there's really four main types of vaccines that are used in people. So one is live organisms, and we're going to go through each one of these types in more detail. Two would be inactivated organisms. Three would be toxoid type vaccines, which are really closely related to subunit vaccines. So first, let's just talk about live organism vaccines, and this really refers to the Jennerian method, where Edward Jenner noticed that vaccinating people with cowpox induced protection against smallpox. And the reason for that is outlined in this cartoon here. If you're exposed, because the cowpox organism and the smallpox organism are related, meaning that they share antigens, if you mount an immune response against the cowpox virus, here you have antibodies that recognize the red or the blue antigens. Some of those antigens may also be expressed on the smallpox virus. And so here you can see that the blue antigen is present on both the smallpox virus and the cowpox virus. And so the antibodies that were induced following cowpox immunization also protect against smallpox infection. And so these are what are referred to as cross-protective antibody responses that can cross-protect against both cowpox and smallpox. Another example of this type of vaccination method using live organisms is using the BCG vaccine that really involves immunization with a bacteria that's related to the Mycobacteria tuberculosis organism that causes TB. So the BCG vaccine is another mycobacterial organism referred to as Mycobacterium bovis, and it induces an immune response that's partially cross-protective against TB, although it really doesn't work all that well and is typically not used in this country. Another class of vaccines that's really related to live vaccines is live attenuated vaccines. And what attenuated refers to is mechanisms that are used to take an organism and reduce its pathogenicity, and use that organism that has reduced pathogenicity as a vaccine that will protect you against the fully pathogenic organism. And so one example of that is the oral poliovirus. And the way that vaccine is made, at least historically, was that the original poliovirus was passaged through non-human host cells, and that caused gene mutations to be induced to allow those viruses to live in a non-human species, not its primary host. And those mutations often resulted in decreased pathogenicity of the virus. And so when you harvest virus after it's been passaged through those non-human cells, that virus often would be less pathogenic and can be given back to people and will induce an immune response that will cross-protect against the native virus. And so this, again, is widely used in the form of a polio vaccine. It's the oral polio vaccine, where you're giving people an attenuated form of the polio virus orally, the natural route to infection. And it's a very effective vaccine that induces protective immunity in the vast majority of people where it's given, and it's also very, very safe. But there is a small risk that these attenuated vaccines may cause harm to immunodeficient patients, because even though their pathogenicity is very much reduced, if you can't mount an immune response at all, sometimes they may cause a syndrome similar to the original infection. The other thing that may happen, at least theoretically, is that the virus may remutate and revert back to its pathogenic wild-type form, although it's not clear that this has ever happened in real life. Another form of a vaccine is, instead of using live organisms, is to use inactivated whole organisms. This is usually achieved by killing the organism either through fixation by formaldehyde or by heating the whole organisms. These are also considered very safe and potentially safer than the live attenuated vaccines, because there's no live organism at all given, unless, of course, you have a mistake in the factory where you've improperly inactivated the vaccine. The immunity induced by these inactivated vaccines is generally thought to be less potent than live vaccines, because it's not the natural infection. They're not able to actually infect cells and induce a natural immune response. The best known examples of inactivated whole organism vaccines that are currently used are the inactivated polio vaccine, or the Salk vaccine, and also the influenza vaccine that we all get every year. These are whole organisms that have been killed to induce an immune response against those organisms. Another class of vaccines can be referred to as subunit vaccines, and in this case, instead of using whole organisms, what they consist of are single or multiple fragments usually of proteins, or other molecular portions of the microorganism, and these can be proteins, peptides, or carbohydrates. Sometimes they're fragments that are cleaved from the organism and purified in the laboratory, or sometimes they're produced using recombinant DNA technology. The advantages of these subunit vaccines are that they often can be produced on a very large scale. It's easy to do quality assurance and assure reproducibility between batches, and because there's no intact organism at all, they're generally considered very, very safe. The problem is, though, because they don't incorporate lots of antigens and they don't incorporate the intact organism, they often have poor immunogenicity without the addition of something called adjuvants and repetitive doses of the vaccine, or multiple boosts. And I'll explain what adjuvants are in just a minute. And so what adjuvants are molecules that often are mixed with subunit vaccines that are designed to enhance or accelerate or prolong the specific immune response to the vaccine antigen. Typically, adjuvants work by activating innate immune cells. They either promote antigen presentation and/or promote the specific production of specific cytokines, and finally, they often act as an antigen depot, or serve to make the antigen relatively insoluble, so it will last in the host for a prolonged period of time to give it the best chance of inducing a strong and potent immune response. There's a variety of different types of adjuvants that are used, although most of these adjuvants are used in research labs and are not used clinically. The main adjuvant that's used clinically is this alum form of adjuvant, which is really just an aluminum hydroxide salt that results in precipitation of the antigen and prolongs its half-life after injection. But it turns out that not only does it prolong half-life, but it also the precipitation of the antigen in that form also activates innate immune cells, largely by activating certain types of pattern recognition receptors associated with damage or danger-associated molecular patterns. So just some examples of subunit vaccines. So one example are these toxoid vaccines that are designed to induce a protective immune response against toxins that are produced largely by bacteria like tetanus or diphtheria bacteria. These toxins often are structurally conserved and are made up of two protein chains with different functions. So one of those chains is involved with binding to specific receptors on cells, and that allows internalization of the toxin, and then the other chain mediates its biologic effect. And what the toxin vaccines usually are toxoids, meaning it's not the intact toxin, it's just the receptor binding portion of the toxin. And then those molecules are referred to as toxoids. They're molecules that have the receptor binding portion, but the toxic arm of the toxin has been inactivated or removed. And what vaccination with these toxoids does is it induces a potent antibody response that will bind to the cell binding portion of the toxin molecule and neutralize it, so it doesn't allow interaction of the toxin with the cell. Another example of a subunit vaccine are vaccines that are designed to induce a humoral immune response or an antibody response to polysaccharide antigens that are part of the cell wall of certain bacteria, for example, Haemophilus influenza or strep pneumonia. These polysaccharide antigens are very bad at activating T cells, but they can engage with B cell antigen receptors and cross-link them and induce a partial humoral immune response, although not as good as when you have T cell help. These kinds of antigens that don't involve T cell help are referred to as T-independent antigens. And so they can activate B cells directly, and then that activation usually involves the B cell producing IgM, but the IgM that's produced is often of relatively low affinity because you don't have a germinal center reaction and you don't have immunoglobulin isotype switching to other immunoglobulin isotypes. And so in order to enhance the immunogenicity of those types of vaccines is that those polysaccharide epitopes are often linked to a protein carrier. And so this is really the same slide that I showed when I was talking about B cell activation, where you have a conjugate of a polysaccharide epitope with a protein carrier, in this case a tetanus toxoid protein. And so the polysaccharide epitope will target B cells that have a receptor specific for the polysaccharide epitope. The antigen is internalized and processed, and peptides from the protein portion of the vaccine will be degraded into peptides presented to T cells, and T cells then can provide help to the B cell. And the antibody that's produced by the B cell will be antibody that was specific for the polysaccharide epitope. So it's a way of providing T cell help to B cells that make antibodies specific for cell wall polysaccharide epitopes that are protective for certain types of bacterial infection. So some examples of conjugate vaccines that are used clinically are the Haemophilus influenza type B vaccine, or the strep pneumonia vaccine, or the Neisseria meningitidis vaccine. All of these utilize this technology where you're hooking up polysaccharide epitopes with a toxoid protein epitope and inducing an immune response that's protective by making antibodies to the polysaccharide. And so let's talk a little bit more about what exactly happens when you're inducing a humoral or immune response following vaccination. So how do vaccines work? And they really depend largely on the induction of antibodies. And antibodies can do all the things the antibodies do that we already mentioned. They can neutralize, they promote opsonization, and also they can activate complement. In part, some vaccines also can activate T cells. This is largely, though, activating the T cell subset, Th2 cells and T follicular helper cells, that are especially good at providing help to B cells. We don't have good vaccines currently that are very good at inducing prolonged effective T cell immune responses against intracellular pathogens that require a CD8 cytotoxic T cell response. And so let's first talk about what's required to generate a protective humoral immune response. So one is that it does require T cell help. And two is you need to have long-lasting humoral immunity that's produced by the induction of long-lived plasma cells that were originally stimulated during the germinal center reaction, left the lymph node, and migrated to other extracellular sites, peripheral sites like the bone marrow, where they continually secrete antibodies of relatively high affinity. And the other thing that helps promote long-lasting humoral immunity is not just inducing long-lived plasma cells, but also inducing long-lived specific memory B cells, so that when they're re-stimulated by that antigen, they can undergo a new or a secondary humoral immune response, undergo additional affinity maturation and isotype switching, and produce additional long-lived plasma cells. And so this is just a cartoon illustrating these characteristics of a humoral immune response that we've really largely talked about already, so I'm going to go through this quickly. So you have a naive B cell and a secondary lymphoid organ that gets stimulated by antigen and by T cell help. These cells will clonally expand in the germinal center. Some of these cells will differentiate into plasma cells and produce antibody, and the first antibody immunoglobulin isotype that's produced during a primary immunization is the IgM isotype. At the tail end of that primary immunization, you'll also start to get some IgG immunoglobulin produced because of isotype switching. But what happens is if you get re-exposed to that vaccine antigen or challenged by infection with that microorganism, you'll get a secondary immune response where you get activation of memory B cells that were long-lived after that initial antigen exposure and expanded in number, expanded in precursor frequency, and you'll get a more robust secondary immune response that's manifested by production of more plasma cells and a lot more IgG produced. And this is really characteristic of a secondary antibody response, where you get a lot of IgG produced. And not only do you get a lot of IgG produced, but that IgG antibody is often more of higher affinity for the antigen because of the affinity maturation events that occur in the germinal center. And so the secondary antibody response, or the response you see after a boost, receiving a second immunization, is really characterized by the reactivation of memory B cells and differentiation into additional antibody-secreting cells upon antigen contact. And as I said, this is predominantly an IgG response with most vaccines. You may also get a secondary immune response upon natural infection, and which is really going to be similar to getting another exposure to the vaccine. Regardless of the way you get that secondary response, you'll get a boost of antibody production, producing a lot of IgG that's very rapidly produced, higher affinity, and is usually protective against infection. And so most vaccines really depend on the production of an effective humoral immune response. It's been much more difficult to develop vaccines that induce a long-lived cell-mediated immune response that depends on cytotoxic T lymphocytes or Th1 effector cells. And this is really thought to be a major issue with the problems in developing an effective HIV vaccine, and also with our inability to develop a vaccine effective against tuberculosis. And it's not fully understood why current vaccines are so bad at inducing long-lived T-cell immunity. But one obvious explanation is that exposure or vaccination with vaccine formulations that include organisms that are either inactivated or subunit vaccines that don't have organisms at all, you don't get live infections. And if you don't have live infections, there's no replication or production of proteins in the cytoplasm of a host cell. As you may recall from the earlier lecture on antigen recognition and MHC presentation, in order to present peptides on MHC class 1, which is required to getting a CD8 T cell immune response, those peptides need to come from proteins that were made within the cytoplasm of the host cell. And since the only way you're going to get that is with a live infection, most of our current vaccines, except for the live attenuated vaccines, are unable to do this. So it's difficult to access MHC class 1 processing pathways with inactivated or subunit vaccines, since these vaccines don't replicate inside cells. And if you don't have access to the MHC class 1 processing pathway, you're not able to activate CD8 T cells and inducing a cytotoxic T effector response. So that's one major problem with why we're not able to develop effective vaccines for all microorganisms, and obviously there's lots of research going on in order to enhance our ability to make vaccines that induce that type of immune response. But the other reason that we don't have kind of vaccines that work for even microorganisms where there are effective vaccines is that we don't have 100% coverage throughout the world. So this is a slide just illustrating the incidence of measles, and you can see that when as the immunization coverage using the measles vaccine increased, the incidence of disease decreased proportionally. But you can also see that there's now a plateau, whereas although 80% of the people in this study were covered, and I think this represents worldwide coverage, there's still a gap of 20% people, meaning that people that are not don't have access to that vaccine are still susceptible to that particular infectious disease. So one of the challenges with vaccination is not just to develop additional effective vaccines that will work on more microorganisms, but also to increase coverage of currently approved and effective vaccines throughout the world. And that's the end of this lecture.